All right, in these next three videos, what we're going to do is we're going to look at the nervous system. We'll start by looking at organization of the nervous system and talk about how we get action potentials. In the second video, what we'll do is we'll look at how two nerve cells can communicate with each other or with other cells of the body, and then we'll start to look at how the nervous system plays a role in movement. And so that's what we're going to do. This first picture just gives you an overview of the nervous system. As you can see, this is what's known as either an organizational or a hierarchical chart. It's hierarchical because we start at the top and work our way down. You know that the nervous system has two major parts, central nervous system and peripheral nervous system. And even though we use nervous system multiple times throughout this chart, it's really all just part of the nervous system. Central nervous system is only brain and spinal cord, and that's because anatomically they're right down the center of the body. The peripheral nervous system is what is to the outside as we get away from the brain and the spinal cord. And there are two parts to the peripheral nervous system, the sensory, which is also known as afferent, A-F-F. E-R-E-N-T, and then motor, which is also known as efferent or E-F-F. And so sensory is information that is coming from the periphery back into the central nervous system. Motor obviously would be commands which are being sent from the central nervous system out to the periphery to what are known as effectors. And there are two branches to the motor part of the peripheral nervous system as well. And so we have the somatic nervous system, again, all part of this overall nervous system, not separate. Somatic would be those nerves that are going to the skeletal muscles. And so they're responsible for voluntary movement. And that's what we're going to focus on as we move along later on. Then we also have the autonomic nervous system, and there are two branches there as well. And so there's the sympathetic branch of the autonomic nervous system. And as you know, this is responsible for fight or flight. And at the beginning of the semester, we talked about how exercise while you're working out really is fight or flight. And so the sympathetic nervous system is controlling all of those changes in the cardiovascular system, the pulmonary system, respiration, endocrine system that are occurring while you exercise. Parasympathetic is sometimes referred to as being responsible for rest and digest. Well, it becomes much more active when you recover from exercise. And so after you stop, the sympathetic nervous system activity decreases quite a bit, and parasympathetic nervous system activity is going to increase and be ratcheted up. Autonomic nervous system activity is sometimes considered involuntary. This picture just kind of shows you the organization of the nervous system in general. And so you can see that we have three basic functions here. The first function is really sensation. Sensation is controlled by sensory receptors. In some cases, receptors are separate cells, but in this particular case, the receptors are just the specialized nerve endings. The blue nerve that you see here is a sensory nerve. In fact, it's what's known as a pseudo-unipolar nerve, and here is the cell body of that guy. We don't have separate sensory cells. In this case, it's just the specialized nerve ending. You can see that the arrow indicates to you that information is being directed back towards central nervous system. Here we have what's referred to as a dorsal root ganglion. A ganglion is a collection of cell bodies in the peripheral nervous system. And so here they are just showing you one cell body. They're not showing you multiple. They're just trying to simplify it for you. But there could be 30, 50, or even more cell bodies here in the ganglion, and that's why it's larger. It has to be bigger to accommodate all the cell bodies that are going to be there. And then you can see that the axon of this neuron is projecting into the back or the dorsal part of the spinal column, and in fact it's here in what's known as the posterior gray horn or the posterior horn of the gray matter. Okay, then we sometimes, but not always, have this neuron which links the afferent and the efferent neuron, and it's known as an interneuron. Sometimes you see it you see it referred to as an association neuron, but I prefer the term interneuron, and I think that's what is most likely used these days. Again, it is here still in the gray matter of the spinal cord, and it's linking the afferent and the efferent. Here is the cell body of the efferent neuron. These are, again, sometimes referred to as motor neurons. This one is going to skeletal muscle, and so in fact it would be an alpha motor neuron, and so it should be heavily myelinated, and it is going out through the ventral root. This is a spinal nerve. So as we get to here, we're in a spinal nerve. And so the spinal nerve has afferent nerves coming in, and it has efferent nerves that are going out. And then as this is the back part of the spinal cord, this must be the front. And so here's ventral. And so the cell bodies of the motor neurons, in particular the somatic motor neurons, are found here in the ventral horn of the gray matter. What we'll see later on is that the autonomic nervous system cell bodies are found here in the lateral horn of the gray matter. That's where the autonomic nervous system cell bodies would be found. Here you can see a typical neuron. This one happens to be multipolar. It's multipolar because it's got lots of dendrites. You know that in general, but not always, dendrites are where information comes into the neuron. Here is the axon hillock, which is part of the cell body or the soma. The axon hillock isn't quite as important as some books make it seem. We'll talk about that in a minute. The soma tends to be large. It usually has a big nucleus with a very prominent nucleolus. The other thing that we find in here, which they're not showing you, would be lots of mitochondria. And then in addition to that, there should be lots of ribosomes and rough endoplasmic reticulum. And so when you see that, you should say, okay, this tells me that these neurons are making lots of protein. That's what they're making. And then here is the axon leading away from the cell body. And generally speaking, the electrical signal is sent away from the cell body going down the axon. Here you can see that these axons will branch down at the end. These branches, which are unmyelinated, are referred to as telodendria. And then here are the synaptic knobs. They're called synaptic knobs or synaptic buttons because you can see, oh look, they swell a little bit. Come back over here and look at the dendrites. No swelling. Here, swelling. And later on, we'll see why that is. You can also see that axons can have collateral branches, which are referred to as collateral axons as well. Again, this axon is myelinated. It's in the peripheral nervous system, and so the myelination in this case is created by Schwann cells. Each one of these little puffy, toasted marshmallow-like things is really created by one Schwann cell. So there would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 separate Schwann cells responsible for the creation of the axons that you see here. Then this important part that we find right here is really the trigger zone. The trigger zone is the area where the action potential is first created or generated, and we'll talk about that in later videos. Here's a different depiction of a multipolar or motor neuron. And so again, the dendrites, the cell body, now they are showing you the mitochondria. Unfortunately, they're not showing you the rough endoplasmic reticulum. And then here is the axon, collateral axon, Schwann cells creating the myelin as we go along. And then those areas where we have bare axon, those are known as nodes, or nodes of Ranvier more specifically. And you can see here that the Schwann cells actually will wrap around the axon in creating those pieces of myelin that we see there. And then again, there's the trigger zone. This is the area where the action potential is first created or generated. Here's a different depiction. And so here again are those bare areas. Those are the nodes in between the pieces of myelination that we have here. There's the nucleus of the Schwann cell. Since it's a Schwann cell, we know that this axon is out in the periphery and not in the central nervous system. There's the axon itself, this yellowish piece. There's the myelinated region, and here you can see again, when we cut through it, we can see it looks kind of like the rings in a tree that we see. And again, that's because the Schwann cell wraps around. Here's a slightly different neuron. This is a pseudo-unipolar neuron. Here are the dendrites over here. I don't think I uncovered that first. Cell body, large nucleolus in there as well. Here's the axon, and the axon actually begins over here. It doesn't begin here, as some books might make you think. And then here are the telodendria, and there, look, synaptic knobs. And then the dendrites are over here. You can see the difference between the ends of the axons versus the ends of the dendrites. And so generally speaking, information is coming in here, and it's moving along the axon toward the cell body and then out to the telodendria and to the synaptic knobs. And in fact, the trigger zone is actually right here, just before we start into the axon. This shows you some very common types of neurons. And again, the top one, there's that pseudo-unipolar neuron, which is a common type of sensory neuron. Again, here's the cell body, which tends to be found in that dorsal root ganglion. That's where they're found. This is a bipolar neuron, and as you can see, rather rare. They tend to be associated with special senses of sight, hearing, and then also smell. And so those guys are more rare. The multipolar neurons are the ones that are most common. And so again, there you have the dendrites. Here's the cell body. And then finally, here's the axon. They don't do a good job here of showing you those synaptic knobs out here at the end of the telodendria. But you can see collateral axons and then telodendria as we branch out there at the end of that. The other thing that tends to happen here is for the multipolar neurons, we've got lots of other axons that are coming in here and connecting to either the dendrites or the cell body. And this is where we're going to have multiple EPSPs or multiple IPSPs, excitatory postsynaptic potentials, or inhibitory postsynaptic potentials. And this cell has to integrate those guys. And that is what determines whether or not we're going to get an action potential here at the trigger zone. And if we do get an action potential here at the trigger zone, it's going to be conducted all the way down along the axon and through the telodendria into the synaptic knobs. Conduction is also sometimes referred to as impulse propagation. Conduction propagation means the same thing, and we're going to talk more about conduction and propagation in video number 2. And so again, integration is where the EPSP's excitatory postsynaptic potentials and IPSP's inhibitory postsynaptic potentials are integrated to determine whether or not we're going to reach threshold at the trigger zone. This isn't a great picture, but it can be kind of instructive. And so again, here are the neurons that make up the peripheral nervous system. And so you look at this one, and I hope by now you're starting to get to the idea where you can say, well, that's a sensory neuron. It's pseudo-unipolar. We're out here in the periphery. We don't have specialized cells making up the receptor. It's just nerve endings. Here is the cell body, which will be located in the dorsal root ganglion. And so what you can't tell from this picture, but what you should know is that the axon is coming in the back part of the spinal cord and ending here in the posterior horn of the gray matter. Posterior horn of the gray matter, where it might synapse with an interneuron. Okay? And so it's hard to tell that in this particular case. Here is the cell body of a somatic motor neuron, and that cell body is actually, again, located here in the ventral horn of the gray matter in the spinal cord, and then the axon heads out and it will go to skeletal muscles. And you can see that we have one axon going all the way from ventral horn of the gray matter all the way to the skeletal muscle. The problem is that this neuron needs to be heavily myelinated for it to be an accurate depiction of a somatic motor neuron or an alpha motor neuron. Always alpha motor neurons that are controlling your skeletal muscles. And then finally here we can see the autonomic motor neurons, and now you can see it takes two to get from the spinal cord to the effector. And now this cell body is located in the lateral horn of the gray matter. It's still to the front, but it's a little bit more to the side, and so we call it the lateral horn of the gray matter, and that's where it's located. Again, in the periphery, a collection of cell bodies is located in a ganglion, and so this is the autonomic ganglion. This neuron is referred to as the preganglionic neuron. Starts in the lateral horn of the gray matter, and axon goes to the ganglion where it synapses with the postganglionic neuron. And so then the axon of the postganglionic neuron will extend out to the effector, which for the autonomic nervous system is smooth muscle or cardiac muscle or glands. And that could be endocrine glands, could be salivary glands, sweat glands, mammary glands, on and on and on and on. Okay, now we need to talk about membrane potentials. Here you can see a depiction of how we measure membrane potentials. We have to have two electrodes. One is out here in the extracellular fluid, and then one has been pushed through the membrane, through the phospholipid bilayer, into the inside of the cell. And here we can see the inside of the cell, compared to the outside, is negative. And so that's what we find. When we look at a typical neuron at rest, we find, actually we find this all the time. Doesn't matter whether the neuron is at rest or not. The concentration of potassium inside the cell is far greater than concentration of potassium outside the cell. And so we've got this strong concentration force that is always moving potassium out of the cell through these things called potassium leak channels. These are proteins. They are always open. Potassium is always diffusing out of the cell. Just the opposite is true of sodium. We have a far greater sodium concentration outside the cell than we do inside the cell. And so sodium is always moving into the inside of the cell. Now, what this doesn't show you is that we have more of these potassium leak channels than we do the sodium leak channels, and so as a result of that, the permeability of the membrane to potassium is greater than the permeability than it is to sodium. It's about 40 times greater. And so there's more potassium leaving than there is sodium that's leaving. The other thing that you need to notice here is that we have these large negatively charged proteins inside the cell, and unlike charges attract, and so that means that not only is sodium going down its concentration gradient, it's also being drawn into the cell by the electrical gradient. Negative charge inside attracts the positive charge, unlike charges attract. It's also trying to hold the potassium in the cell, but the concentration force is so great that potassium still leaves. Potassium always tries to leave. This shows you a slightly better depiction of the leak channels. In this particular case, the potassium is represented by the orange diamonds. More potassium leak channels than there are sodium leak channels. Sodium is represented by the gold balls. But in either case, potassium is always leaving, sodium is always coming in. And then the other thing that we see here would be the sodium-potassium pump. They're constantly trying to move the sodium out of the cell and the potassium back into the cell. Potassium is always, always, always moving out, and it's trying to reach equilibrium. And the only time it would reach equilibrium is when the electrical force across the membrane balances the concentration force. And so again, unlike charges attract, and that means that the force inside the cell has to get all the way down to -90 in order for the concentration force to be balanced by the electrical force. And so the potassium equilibrium potential is -90 millivolts. Here we can see resting membrane potential. Here's our electrode in the extracellular fluid. Here's the electrode that's been pushed through into the inside of the cell. This cell is at rest. Unfortunately, they're not showing you leak channels or sodium-potassium pumps, but that's the case. Sodium is always going to move into the cell until it reaches equilibrium, and for it to reach equilibrium, the inside of the cell has to become positive 65. And you can see that this needle has to move all the way over towards here, which it never does. And so we never reach the sodium equilibrium potential. But it would have to be positive inside in order for the positive charge inside to repel the positive charge of the sodium ions trying to come into the inside of the cell. Again, more potassium leak channels than there are sodium leak channels. Potassium is leaving. It's trying to get to -90, but because some sodium is coming in, it's keeping the resting membrane potential close to that -70 as we see right here. Terms you need to be familiar with. You need to understand what depolarization means. Depolarization means that the inside of the cell is becoming less negative. Again, here we can see our setup before we've put the recording electrode into the inside. Both electrodes in the extracellular fluid, it's just going to read as negative millivolts, no charge whatsoever. Now we push the recording electrode through the phospholipid bilayer. Oh, look at that. We get down to our -70, and so now we can see. In order to depolarize, what we have to do is we have to have the inside of the cell become less negative. Even if it went from -70 to -60, that's still depolarization. -60 to -50, more depolarization. We can go all the way up past 0 and become positive, but we don't have to. And so even moving from -70 to -60 is still depolarization. Repolarization means we're going to move back towards -70. Typically, it always means that we're going to go from -60, -50, +40, back towards -70. On the other hand, what we can do is we can make the membrane even more negative than -70. That's what we've done here. Look, we've gone down to -90. We've reached the potassium equilibrium potential. And so if we make it more negative, then we've hyperpolarized. Technically speaking, if we go from -90 to -70, you could consider that repolarization, but we normally don't mean that. And so that's not typically the case. Well, how is it that we change this membrane potential from -70 to whatever? The only way we can do that is by making the membrane more permeable to the different ions. And the way we make it more permeable is through ion channels. That's what we do. If we're going to depolarize, we have to let positively charged ions come into the cell. That's what we have to do. And if we're going to repolarize, we have to let positively charged ion leave. And here you can see, look at that. We've opened a sodium channel, and as a result, sodium is coming in. Positively charged ion is coming into the cell, depolarizing it, making it less negative. Here you can see an incredibly important ion channel that you have to know. This is a voltage-gated sodium channel. Normally it's closed, and there are two parts here, an activation gate and an inactivation gate. At rest, the inactivation gate's open, activation gate is closed. We stimulate it, the activation gate opens, sodium goes down its electrical chemical gradient into the inside of the cell and starts to depolarize it. Here's an even better depiction of the voltage-gated sodium channel. Here it is, it's closed. Sodium enters, but it can't get all the way through until we've opened the thing up completely. I like this because you have to remember these are all made out of protein. These things are schematic representations that you see here. This is a little bit more realistic. Remember the secondary and the tertiary structure of proteins. That's what they look like. They look like these Christmas ribbons on packages. And then here we're looking from the top down. There's that opening, which at rest will be closed. The sodium can't get through. But once we stimulate it, it opens up, sodium goes in, we start to depolarize this guy. And then to repolarize, as we said, we've got to let positively charged ions move out of the cell. That would be the potassium. Positively charged potassium. I didn't mean to cross out the potassium. And so now we also have voltage-gated potassium channels. When the voltage-gated sodium channel is shut and the voltage-gated potassium channel is open, we repolarize. We move back towards the negative 70 millivolts that we're trying to get to. Here you can see the graph that shows the action potential. And I want you to go ahead and look at the two axes. Pay attention to them. Pause the video, look at them. And so you should have said, oh, look at this. The y-axis is membrane potential, it's voltage, and the units are millivolts. Very, very, very small units. Think of a 9-volt battery. It's that little square battery with the two poles, positive and negative, both on top of the battery. That's 9 volts. Well, this is millivolts. Far, far, far less than that. And then the x-axis is time in milliseconds. This action potential is complete in less than 5 milliseconds. Very, very, very fast as it's happening here. Here you can see that the dark dashed line is the resting membrane potential, negative 70 millivolts. And here you can see that the red line is what we refer to as threshold. We have to get to threshold in order to cause the action potential to occur. Okay, and so at 1, we've opened up voltage-gated sodium channels. Sodium is going down its electrochemical gradient. It's entering the cell and it's causing depolarization. At this point, up at the top, there you can see a different depiction of the voltage-gated sodium channel at rest. When we stimulate it and we open up the activation channel, and then when the inactivation channel has shut up here at the peak, and that's what's going on at 3. The voltage-gated sodium inactivation gate is shut right over here. Sodium can enter, but it can't get all the way through. But at the same time, the voltage-gated potassium channels now are open. Potassium is leaving the cell, and that's causing the membrane potential to be driven back down towards negative 70. But you can see that, in fact, the potassium is leaving so fast that we tend to overshoot here. And here you can see that at the same time that we open up the voltage-gated sodium channels, we open up the voltage-gated potassium channels. They respond more slowly. They're not fully open until we get over to here roughly. Okay? And you can see that because they don't shut right away, we actually get this period of hyperpolarization. Then the voltage-gated potassium channels are shut. The sodium-potassium pumps are furiously pumping away, getting sodium out of the cell and potassium back in. And finally, we're back to our resting membrane potential of negative 70 millivolts. And so lastly, what this is showing you is the blue line again shows you that action potential. Look, resting membrane potential, threshold, depolarization. And these two lines are showing you the membrane permeability. The green shows you membrane permeability to potassium, which is higher at rest than membrane permeability to sodium. And here it's kind of the brownish line that you can see here. And so as the voltage-gated sodium channels open up, the permeability to sodium goes way up. Sodium enters the cell, and we get that steep, rapid depolarization. At the peak, The inactivation gate of the voltage gated sodium channel is shut. No more sodium can come in. The voltage gated potassium channels are open. So look, the permeability to sodium has dropped way off. The permeability to potassium is at a peak, and so we get repolarization. And then finally, all the channels shut. Unfortunately, we've little bit too much potassium leave the cell, and so we're down near the potassium equilibrium potential of about negative 90 millivolts. Now those sodium potassium pumps have to pump the sodium back out and the potassium back in until we get back to negative 70 millivolts. And that brings us to the end of video number 1. And in video number 2, we're going to go ahead and look at how that action potential makes its way down to those telodendria and the synaptic knobs, and how we communicate with other cells.